Hi, I'm Jamie Bax, your instructor for A&P2. We're going to continue on with our discussion of renal physiology. And let me switcheroni here, screen share mode. That's where we left off. Let's talk about what happens in the renal corpuscle, the actual sequence of events here. So you have your afferent arteriole, which is bringing the blood into the corpuscle. Remember, afferent arteriole branches from the renal artery. The glomerulus is this big capillary network, which is where filtration occurs. The efferent arteriole is taking the leftover blood that's inside of our capillaries out of the glomerulus and recollect stuff from the peritubular capillaries. The capsular space is the space between the glomerulus and that capsular wall. So the corpuscle is the whole thing. You have your glomerulus and then this big catcher's mitt surrounding the glomerulus, which is the capsule. The capsular space is the space between the glomerulus and that outer capsule. There is something called a JG complex or juxtaglomerular complex. Sorry, I thought that was my daughter. This is a small little network of cells. It's right next to the afferent arteriole and the DCT. This is the structure that's going to kind of check into the blood and make sure that there's enough red blood cells in our blood. If there is not enough red blood cells in our blood, or if there's not enough blood pressure, not enough volume, renin will be secreted. Renin is going to work in our angiotensin, aldosterone system to increase our blood volume and blood pressure. So there's also mesangial cells. These mesangial cells are supporting cells. These are cells that are basically next to the glomerular capillaries, and they're controlling the flow and the diameter of the capillary blood flow. So that, and you'll see that the flow of blood through the capillary is going to affect how efficient our capillary is at filtering. So here you see this entire image of the afferent arteriole coming in. You see our glomerulus itself covered with these little cells. The cells are called podocytes. Is that written on this? Kind of, yeah, towards the bottom. Podocytes are the specific names of the cells of our glomerular capillary. You see the capsule itself surrounding this capillary network, and the capsule kind of acts as a catcher's mitt, catching everything, leading all of the filtrate to the PCT. You also see the efferent arteriole, and in between the efferent arteriole, the afferent arteriole, and the DCT is this JG complex, where there are special cells which are tasting and getting in touch with the blood to make sure there's enough blood volume, and therefore adjusting hormone levels so that our blood pressure can be elevated. The mesangial cells are within our glomerulus, kind of holding everything together and working to adjust the diameter of those capillaries. So let's talk about the capsule. So the capsule has this outer layer to it, has a parietal and a visceral layer. The parietal layer is the outer capsule, visceral layer is the inner capsule, and there are podocytes within the glomerulus. Those are these large kind of blobby-like cells. They wrap around all of our glomerular capillaries. The gaps in between the cells, so the cells are kind of fitting around each other like that, and then there are gaps or spaces in between. Those are called the filtration slits. The filtration slits are the sites of filtration. So you can imagine that the blood flowing into this glomerulus is going to, hit the walls of the capillary, and anything that can leave will leave through those filtration slits. Materials will pass out of that capillary, but they have to be small enough to fit through those filtration slits. So like we mentioned, protein, nope, not going to fit. Lipids, nope, not going to fit. There you see those lovely little podocytes and the filtration slits in between, kind of where they are situated. The membrane itself, the membrane that our substances have to go through, there's three components to this membrane. There's the fenestrations which are within the glomerular capillary. So this is the actual lining of the capillary itself, right? So in the blood, we're talking about like the tunica intima here, right, of our capillaries, and it's just one cell thick, and these are fenestrated capillaries. These fenestrated capillaries, if you remember our types of capillaries, fenestrations are very large holes, not as large as sinusoids, which are in the liver. They're smaller than that. However, they're still relatively large compared to the continuous capillaries that are spread throughout the body. Then there's a dense layer. This dense layer is basically the basement membrane gluing that inner lining of our blood vessel capillary to the podocyte. So the basement membranes in between. The podocytes themselves cover this whole network and form these filtration slits. So in order for stuff to get from the inside of our blood to the capsular space, it has to go through the fenestrations of our glomerular capillaries, and then through the basement membrane, and then through the filtration slits. Got it? So all of these layers are meant to prevent things that shouldn't be leaving the blood, like plasma proteins, into that capsular space. Here you see a much better picture of what everything is in terms of this filtration membrane. You see that inner capillary lumen are fenestrations or pores of our capillary, that dense layer, and then the filtration slits themselves formed by the podocytes. So let's talk about how this rate of filtration is controlled. It's all regulated by a series of feedback loops. One of the major factors that's controlling glomerular filtration is the glomerular hydrostatic pressure. This is the pressure, the blood pressure, within the glomerular capillary itself. Hydrostatic pressure, if you remember back to our capillary dynamics, hydrostatic pressure pushes stuff out. It pushes stuff out of the blood and into the interstitial fluid. So the glomerular hydrostatic pressure is pushing water and solutes out of the plasma into the filtrate, into that glomerular capsule, right, into that capsular space. It's higher in this area than in other areas because of the different diameters of our afferent and efferent arterioles. Typically, our afferent arteriole is very much dilated. Our efferent arteriole is smaller. So this is going to slow the flow of blood through our glomerulus. That slow flow is going to increase the hydrostatic pressure. With an increase of hydrostatic pressure, you're going to kick more stuff out at the glomerular capillaries than you would at, say, the capillaries in your brain or anywhere else in your body. So the capsular colloid osmotic pressure is the amount of pressure in that capsular space. It's almost nothing. So in other words, this hydrostatic pressure is pushing water and solutes out of the glomerular capillaries into the capsular space that pretty much doesn't have any pressure at all, right? There's no pressure pushing anything back in. It's just taking that pressure out. Because as soon as everything's kicked out of the glomerulus, it's just going to go right into the renal tubule, right? It's just going to go right to the PCT. It's not going to hang out in there for a while, right? There's also blood colloid osmotic pressure. This is the pressure due to the materials in the solution. This is drawing water out of the filtrate and back into the plasma. This is the opposing force to filtration. So in order to create filtrate, you have to our glomerulus has to overcome blood colloid osmotic pressure and capsular colloid osmotic pressure. Capsular colloid osmotic pressure is almost nothing. Blood colloid osmotic pressure is minimal. So this buildup of hydrostatic pressure in our glomerulus is going to cause a net movement of fluids, right, water and solutes, out of the glomerulus and into that capsular space. There's also capsular hydrostatic pressure, which is opposing our GHP, our glomerular hydrostatic pressure, tending to push some of the water and solutes back into the plasma as well. This is a result from the filtrate that's already there, right? That's kind of sitting and waiting to go into the renal tubule. It's relatively small here as well. So overall, the net filtration rate is pushing water and solutes from the glomerulus, the blood in the glomerulus, into the capsular space. So there's always this net movement out of the blood and into that capsular space and then into the PCT. So net filtration pressure is this kind of balance of the pressure across our capillary networks. It's the sum of all of our hydrostatic pressures and colloid osmotic pressures. It's right around 10. Again, you don't have to know numbers. You just have to know that typically the net filtration pressure is positive. Right? We're pushing stuff out. Average pressure, that's the average pressure of pushing that water and solutes out of the glomerular capillaries into that capsular space. Here's a good schematic view of how all that stuff works, right? So you have your overall glomerular hydrostatic pressure, highest number on there, pushing everything out, right? You have minimal blood colloid osmotic pressure, pulling it back in. A little bit of capsular hydrostatic pressure, putting back in. Capsular colloid osmotic pressure, practically zero. There's nothing else pushing it back in other than capsular hydrostatic pressure. So we have a net filtration pressure of around 10, pushing it all out into that capsular space and then into the PCT. You can pause the video here. In fact, I encourage you, because your head's probably spinning right now with all of these terms, pause the video here, review these questions, go back and re-listen to what I just said. Right? That's one of the beauties of having this course recorded for you. You can go back and you can re-listen to everything. And I know I talk fast, so you probably do that already, hopefully, but go back, re-listen to everything, see if you can get a good grasp on all of these terms and how they relate to one another. We're going to keep moving and looking at how our GFR, or glomerular filtration rate, is created and adjusted. So the GFR, or glomerular filtration rate, is the amount of filtrate that's produced by each kidney each minute. Right? This is similar to our functional expiration volume, right? Or our cardiac output, right? This is just looking at how much are we actually producing in a given time period. And so it's just looking at the amount of filtrate or urine in a minute. Each kidney has a huge amount of filtration surface, right? Over 64 square feet, you don't have to know that number, of filtration surface in this little tiny kidney. So on average, we're around 125 milliliters per minute, which is around 180 liters per day. Remember how I said if you were to not reabsorb anything from the glomerular, after the glomerular filtration, how much you would have to pee. I think I said 8 liters. I was way off. 180 liters. I knew it was something astronomical. Most of it, 99% of it, is reabsorbed. And that makes sense, right? Because we're not peeing all day long. If you are, there could be a problem, right? So how is it controlled? Everything in our body has 3 levels of control. Auto-regulation at some local level, endocrine control, and hormone and neural control. Right? So that's all done centrally. The endocrine response is initiated by the kidneys through renin, right? Through other local hormones, EPO. Our neural response is all involving sympathetic. Our kidneys are under sympathetic innervation from our autonomic nervous system. Here's how it all works. You have some type of decrease in GFR. So you're going to, limit the amount of, there's some limit for some reason on the amount of filtrate that's being produced at the kidneys. Maybe you're dehydrated, right? Maybe, for some reason you just haven't been drinking enough water and your kidneys output way down. So the afferent arterioles are going to dilate. The mesangial cells are going to contract. This is going to increase the amount of blood that's being passed through the glomerulus and therefore into the capsular space, increasing. Bye, Heathers. Sorry. The efferent arterioles are going to constrict. This is holding the blood in that glomerulus for a longer period of time. This is going to increase GFR, or glomerular hydrostatic pressure, ultimately increasing GFR, but we're not there yet. So all of those things happening in the glomerulus are going to cause an increase in glomerular blood pressure. Once that blood pressure gets up in the glomerulus, it's going to increase glomerular hydrostatic pressure, which is going to push more of that blood out, which is going to get your glomerular filtration rate back up to normal. So here's the glomerulus again. Our JG apparatus, juxtaglomerular complex, this is the key in regulating all of this, because that guy right there, right at the efferent and afferent arterioles, is going to be responsible for contracting and dilating our efferent arteriole, regulating this GFR rate. If we look at the opposite scenario, or actually just kidding, let's look at the same thing, right? we have, but we're going to look at the central nervous system regulation. Our endocrine response here, when we have some type of change in our GFR, our JG apparatus is going to release renin. Renin is going to trigger this angiotensin-aldosterone system. Angiotensin goes up and is converted into angiotensin, renin is released and is converted into angiotensin 1 in the liver, and then that goes into our lungs and is converted into angiotensin 2 in our lungs by angiotensin converting enzyme ACE. Angiotensin 2 then goes down to our adrenal glands and releases aldosterone. Aldosterone is going to increase our rate of sodium retention. Because we're holding on to sodium, we're therefore going to hold on to water. So we're going to increase fluid consumption as well, increase fluid retention. Constriction is going to occur in the venous system, and overall increase our cardiac output. With an increase in blood flow and an increase in blood volume, we're going to increase blood pressure, getting that glomerular pressure up, which is therefore going to get our glomerular filtration rate up. Did I go too fast? Listen again. All right, you can pause the video. Actually, you know what? I think I'm going to stop the video here and go back and review this section with these 3 questions so that our next little section is on a slightly different topic here. All right, bye.